Thank you so much. So, ladies and gentlemen, we are honored to be here. First of all, on the slide, you can see Tammy. Tammy is an AI-generated figure who helped us through the process. Our presentation is about skin itching, the factor of skin itching, which is histamine, and our solution to this problem. We are high school students from Szeged, Hungary, eager to learn molecular biology and being aware of our school's proud history of participating in iGEM, we urged our biology teacher to initiate a new iGEM project, allowing us to showcase our skills and creativity in the field. For working effectively, we used Scrum management, led by Scrum Masters and supported by product owners. Initially, we set out to address skin itching, also called pruritus, by targeting histamine as a key factor. While previous iGEM teams focused on oral histamine using DAO enzymes, we developed a topical ointment that combines DAO and HNMT enzymes and DNA aptamers to reduce histamine levels in the skin.
As we designed our project and experiments, we thought of optimizing the histamine metabolizing enzymes HNMT and DAO for E. coli as a contribution to the work of previous iGEM teams, for which we have uploaded to their respective registry part pages. We have also contributed to the Lipid Handbook of Linkoping this year. We split our experimental plans into two series of experiments we named upstream and downstream. Because of the time available, we carried out these two sets of experiments in parallel. In our upstream process, we have located the coding DNA sequences of HNMT and DAO in the NCBI database. We then performed codon optimization on the sequences and designed their appropriate ligation into the PET-duet1 vector. We sent these sequences to synthesis to the firms IDT, GenScript, and Twist Bioscience. After the arrival of our DNA, we started the process of amplifying the sequences by PCR. After several rounds of experiments, we have managed to design adequate protocols for the reactions by refining our mixture of reagents. We have successfully transformed our bacteria with the heat shock method, and we purified our proteins. We proved our results by performing a vertical page electrophoresis and an ELISA assay. We have found that projects generally use the SELEX process to find suitable aptamers. However, we have seen several pieces of scientific papers listing suitable sequences for histamine. Therefore, we have chosen 3 sequences from 3 different articles. We have also found out from our discovery of aptamers whether they can be used for skin diseases. Fortunately, in September of this year, a new scientific article announced the successful usage of aptamers in specific skin diseases. This news motivated us to continue our experiments. We planned our aptamer collection in 2 different compositions. One, to measure fluorescence change to assess the change in the presence of histamine. By attaching a fluorophore to a 5' end of an aptamer and a quencher molecule to a 3' end of a complementary DNA strand, fluorescence can be controlled. When the complementary strands bind to the aptamer, it prevents the aptamer from folding and binding histamine, therefore resulting in low fluorescence. However, in the presence of histamine, the quencher strand detaches, allowing the aptamer to bind histamine at increased fluorescence, thus allowing the detection of the binding of histamine. Our experiments proved that fluorescence is a relevant way to assess aptamer efficiency. The drawback of the technique is the expensive fluorimeter, which is not present in each laboratory. The other composition was created to offer a sustainable tool besides usage for topical methods. The method's essence is that the streptavidin on the surface of magnetic beads can bind biotinylated single-stranded DNA, the sequence of which is reversely complemented to the elongated part of the aptamer. When we apply a strong magnetic field, we expect to gather not only the beads, but also the elongated aptamers. These aptamers can trap the histamine, bind to the magnetic beads connector sequence, and thus purify histamine itself from the liquid. As our budget could not cover an enough sensitive histamine assay to test our idea, we decided to do it in 2 parts. Firstly, we successfully proved that these beads could attach the elongated aptamers, and secondly, we proved that the elongated aptamers could bind histamine successfully. We proved that the aptamers can efficiently bind histamine when the concentration is below 10 micromolar.
In our downstream process, we check the transfer of our enzymes and aptamers into the skin with two types of transdermal devices. We performed some experiments and measurements at the Faculty of Pharmacy at the University of Szeged. We used liposomes to package our enzymes and aptamers, and we used hydrogel as a carrier media, and we measured the transfer of our ingredients with and without packaging them into liposomes. The transdermal devices were running for 24 hours. Using the ex vivo transdermal device, we proved HNMT can be transferred into the skin with liposomes and hydrogel media, but the amplification time is 6 hours or more. This suggests HNMT could be worth using in topical treatment, but mainly in patients with chronic pruritus symptoms. The ex vivo transdermal device didn't release the DAO enzyme neither in liposomes nor without it, making the HNMT a better choice for topical use. Our aptamers didn't enter the skin in the used hydrogels, but according to our previously mentioned experiments, they can still be efficient ingredients to decrease histamine levels in food.
We followed all personal, environmental, and data safety rules. The Department of Pharmaceutical Technology helped us by testing our active ingredients with an ex vivo skin penetration device. They carried out the experiments with approval from the Hungarian Medical Research Council. This device uses human skin samples from surgeries, therefore consent forms were signed by the patients.
We collaborated with numerous experts on their field to help us make our product better and more desirable to the market. Moreover, we had the chance to work with the Faculty of Pharmacy at the University of Szeged, which was incredibly helpful to make our measurements as accurate as possible. We knew we will be a small business at first, therefore we contacted the local business owner, Edina Pallagi. With her experience in the cream development field, she could highlight parts of our project that we had never thought about, making it more corresponding for the real market's challenges. Another fancier was Dr. István Kiss, who's the director of a biotechnological institution in Szeged. Apart from the knowledge that we gained during the meetings with him and asking him questions, we could see the equipment which we'll need to produce our active ingredient, like fermenters, in their real size. We analyzed the informations that we gained during these meetings and utilized them. We thought it's also negligible to help our potential customers decide their levels of itching. Therefore, after consulting with medical professionals and the current scientific literature, we developed the pruritus questionnaire to assess their grade of itching. This questionnaire consists of two parts. The main part contains general questions about the level of itching a person might have, and the second part refers to special conditions such as dialysis and diabetes. As we were not allowed to enter the local hospital to interview real patients, we asked two former iGEMers to do these interviews for us. They recalled the information on 17 patients, which was the first systematic examination on pruritus in this department. These questions make a comprehensive view of the patient's level and intensity of itching and the impact on their lives, evaluating the answers given.
In our education project, we targeted two different levels of the Hungarian education system, primary schools and secondary schools. We have given several lectures on synthetic biology for primary school students. The main part of our lecture was a Lego game. During the game, players have to build up a DNA model and go through the steps of central dogma. To test how understandable our presentation was, we filled in questionnaires by the students before and after our presentations. The results are shown in this chart. As you can see, there were improvements on almost all the questions. It was a great pleasure to see how enthusiastic were the children about playing the game. We also developed a video game for secondary school students. The aim of the game is to create an opportunity for students who aren't given the chance to work in an equipped laboratory and try out different laboratory techniques such as PCR. As you can see, we sent the link of the video game to several secondary schools, and it has reached more than 500 students from 67 institutions across the country.
We've created models for enzyme diffusion through the skin, for the velocity of enzyme reaction, and for the decrease in substrate concentration by the velocity of enzyme reaction. The solution for these mathematical equations are the graphs which showcase our product's feasibility for topical use. As the founders and leaders of business society in our school, we decided to create a business plan for our iGEM project. There are many opportunities for biotechnological companies in Szeged. This is shown by the numerous well-known companies with headquarters here. Our company will produce active ingredients, meaning our enzymes and aptamers. We would apply for venture capital funding and loans as a starting capital. The company would be built in the industrial area of Szeged with the help of local construction businesses. We would search for Hungarian human resources, mainly for the expertise and low salary needs. Our procurement would be based on local and reliable companies who we plan to create long-term partnerships with. Our potential customers are medium and large-size cosmetic and pharmaceutical companies who we plan to reach through tailored business offers and industry conferences. Our main threats are financial dependency, delays in procurement and production, and regulatory problems when entering new markets. We would cope with these problems through long-term partnerships, rigorous production planning, and with the help of experts, we will monitor all changes in our target markets. Our potential exit strategies are private equity buyout and partnerships or acquisitions by larger companies.
Sustainability is essential for our future, and it has become a call sign in the 21st century. Our work at the Sustainable Development Team was based on two pillars. Firstly, the Sustainability Profile Assessment Tool, or SPOT for short, and secondly, our 3D printing solutions. SPOT is a questionnaire which measures a company's sustainability in 8 different factors, like supply chain sustainability, waste management, social responsibility, and so on. The results can be evaluated easily, and they can be displayed on a radar diagram, which gives the companies clear and straightforward feedback on what aspects they need to improve. The second part of our work was to examine the potential use of 3D printing in the field of cosmetic packaging. During our work, we also produced a 3D printed jar. The chosen material was PLA, or polylactic acid, because it's made from organic compounds like cornstarch or sugarcane. The jar can be easily refilled with the ointment, or if it's not an option, it is also biodegradable. At the beginning, we had concerns about PLA, as it lets substances containing large amounts of water through. Because of this, we carried out experiments with creams and PLA plates of different thicknesses. Luckily, none of them let cream through, and we used the gained experience in the design of the jar. At the end of our presentation, we would like to thank our sponsors for their financial support, our teachers and the university staff for their help, and we would like to thank everyone else who had contributed to our work. Without them, our project could not be possible. And finally, we would like to thank you for your attention.
Thank you for this very impressive presentation and the excellent timing. And now I ask you one by one to come forward and receive your certificate. First I ask Emese Bakos. Next, Csongor Rihárd Orvosi. Tamás Nagy. Norbert László Nyeste.